For the second part of the Biomineralization and Taphonomy lecture, of course, as the slide says very clearly, we'll be talking about taphonomy today. So taphonomy is an incredibly fascinating subject that has a lot of broad applications to how we understand the fossil record. And the basic definition of taphonomy is everything that's happened to a specimen between death and fossilization, which includes scavenging, decay, transportation, so wind or water moving stuff, erosion, weathering processes, burial. Now we're going to wrap up taphonomy into the broad, just using the word taphonomy here, but the fossilization process is slightly separate, and that's the process of converting living tissues into rock after burial. So this could be physical diagenesis or chemical diagenesis. The definition of a fossil, we actually do have a very particular definition of a fossil. So if you find a squirrel on the side of the road that recently died, you wouldn't call that a fossil, right? Well, when would you call it a fossil? The general age is 10,000 years. So it's not like we sit around waiting 999 or 9,900. I give up. It's not like we sit around waiting for that 10,000 year mark to start studying things. So the looser definition, the broader definition, which I like better, is once we start to see any diagenesis is when we typically call it a fossil. So taphonomy is incredibly important because it can tell us a lot about how an environment was when the organism was alive. It can tell us about how it was buried. It can tell us about the geologic history of the rock. It can tell us about chemicals that were present in the area. And it can, most importantly, help us see the biases present in our study. So taphonomy gives us a window into the fossil record. The more we understand about how fossils are preserved, the more we'll start to understand what we can and cannot see and what we can and cannot study through the fossil record. We'll talk more about that in a second. But intrinsic biases, these are features of the organism itself, that can affect its preservation potential. So for example, size, robustness, how strong are the bones, the number of elements to the body, and the chemistry of the hard part. These are the intrinsic biases. So think about it this way. If you are thinking about going to look for fossils, what do you think you're going to find? Do you think you're going to find bones? Do you think you're going to find soft tissue? Well, as we talked about in the last lecture, we're much more likely to find bones. So these are things that are intrinsic biases. The jellyfish cannot help that its fossil record is terrible, just as clams cannot help that their fossil record is awesome. All right, so size here. Size can kind of go both ways. And what I mean by that is these tiny little fossils here, these are radiolarians down in the bottom left-hand corner, gorgeous fossils. They're made out of silica. All right, so they're hydrated silica. These are excellent fossils. They're so tiny. They're actually not that difficult to preserve. They kind of just get washed up and deposited in mud. Now, the Komodo dragon is a lot larger. Its bones are not as likely to be found, or if they are found, only a few of them are typically found at a time. So this is a varan, I believe it belongs to the varanid group of lizards. I might be wrong on that. But Komodo dragons are not as easily preserved as radiolarians. They're going to preserve in a much smaller number. However, thicker bones, more robust bones, can also be a better preservation material. So for example, if we're talking about things like pterosaurs, the flying pterodactyls, right? Pterodactyl is just one genus. Pterosaur encompasses the whole group. They have hollow bones. So if we compare that to, let's say, Apatosaurus, the long-necked dinosaur, which has non-hollow bones, very thick, the Apatosaurus, even though larger, will be much more likely to preserve. So there's a couple different biases at play when we talk about science. Size. There we go. Robustness. So here are two clams, bivalves, mollusks. Those are their names. All right. Bivalvia belongs to phylum Mollusca. And as you can see on the left, these shells are incredibly thin. This is a mollusk that likely lives in a lake. This is a freshwater mollusk, and its shell's incredibly thin. Think about how this might play into where it would live. If you had an incredibly thin shell, would you want to live in quieter water or higher energy? You'd probably want to live in quieter water. So that's great for living, but for paleontologists, it's not going to give us the best fossil record. Freshwater mollusk fossil record is not that great, and primarily because they've got very thin shells. This is in contrast to this robust beast of a clam here on the right with incredibly thick shells. This is going to be much more likely to preserve in the fossil record. Number of elements. Let's compare the crinoid, this beautiful, beautiful, beautiful fossil, to this humble snail here. The snail has one hard part to it. That's it. You're looking at the shell. The crinoid has thousands. So if you look here, every single one of these arms is composed of hundreds to thousands of teeny tiny little plates, arm plates, that will disarticulate within moments after death, quite literally hours. So the average starfish has over 1 million pieces to its body. That's a starfish. This is a crinoid, of course. This has hundreds of thousands up to 1 million. All right. So as you can imagine, the only way we're going to get a fossil like this is to catch it before it dies. And how are we going to do that? Well, we're probably going to bury this alive. This represents a storm burial event where all of these crinoids were still alive when they were buried and their journey as a fossil began. Chemistry of hard parts. We've talked about this a little bit. We know about the different fossil-forming minerals. We have calcium phosphate, which is bones and teeth. We have calcite, aragonite, and we have hydrous silica. As you might remember, aragonite typically is where we find aragonite in things like mollusks, snails, and bivalves here, which is what we're looking at. If you remember with me, this mother of pearl sheen is called nacre. That's nacre microtexture of aragonite. But we know aragonite isn't as stable as calcite is, so this lowers its fossil forming potential. Now there's another category of bias here, extrinsic biases, features of an organism's environment that can affect its preservation potential. This could be abiotic or biotic, meaning non-living or living factors of the environment. It really just depends. So extrinsic factors, the environment itself. So before we move on, to that next one, let's talk about some of those biases. Let's see. If you die in an area that is highly scavenged, you may not have the greatest fossil record, right? If your body is scavenged. If we find organisms dying in the middle of a very humid environment, we can probably guess that that's not going to give us a great fossil forming potential, because humidity can often cause more organic decay. So there's a couple different things that we will look at here before we move forward. So here is taphonomy as a whole to talk about when we're going to mix together the extrinsic and intrinsic biases here too. So here we have our friend the panda. This is a real paper that was published. And what it was trying to figure out how to describe how fossil pandas got caught in the lower chamber of a cave. So here we have a beautiful story, which is kind of sad being painted. We have our panda here minding their own business, trying to get some bamboo, living its best panda life, when all of a sudden tragedy strikes. It falls. It does a tuck and roll. Somehow it then also becomes dead in the exact same position in the next panel. It gets washed away by river. This would be an extrinsic bias here, right? It leads the bones to being disarticulated and washed into a river. And then finally it's deposited in this nice little cave. You can see the stalactites here. These are likely calcium carbonate stalactites, just from my knowledge of caves. This could be another extrinsic bias because it's dripping chemical-filled waters on top of the organism that might affect it. Its intrinsic biases here might be that pandas have a lot of skeletal elements, a lot of bones. It's going to be nearly impossible to get every one of these bones preserved. They have a larger size, also limiting what we can see in the fossil record. So let's talk about each of these in detail. So the best and worst part about taphonomy is it can be pretty gross, right? So one of my graduate school jobs was working for a taphonomist who studied the bite marks of alligators. So what she would do is she would throw old cow bones from meat factories into alligator containments And then she would watch them eat whatever was left on those bones, and she would study the bite marks afterwards. So my job was to scrub the nasty bones after the alligators had eaten them. It was a horrifying job, and it is pretty much what convinced me that taphonomy was not for me. So if this is something that you're just like, wow, I really need to do this, I know some people. All right, so predation and scavenging. This is often the first thing to happen after an organism dies. And as you know, because we live in an area with a lot of roads and a lot of wildlife, this happens quickly. So we have turkey vultures here in Tampa. We have other scavengers that will quickly disarticulate the organism and badly damage them, take away bones and things like that. Here's an excellent video of this. This is pretty horrifying. So this is a time lapse of ants eating a gecko, so you can see how this works. The buildup is pretty intense. My apologies. All right. So one of the best things about taphonomy too is that we can really see this happening in real time. This is what gives us an actualist experiment. We can start to run our experiments on recently dead organisms to understand how fossils are made. So our poor gecko friend is now being carted off into multiple pieces. I will not show the wild elephant being scavenged, but there is a documentary of it, and you can watch it on your own time if that's something that you want to do. But now you see the bones being carried away. This is literally just hours after death. So as you can imagine, the fossil record is filled with the winners of those who were not scavenged, really, right? This gecko would never make it as a fossil. It's being carted off into tons and tons of tiny pieces. The bones are very small, delicate, multiple bones. There goes the skull. Holy mackerel, carrying away the skull. Ants are absolutely wild. Arthropods are just very upsetting in general, aren't they? All right, so let's stop watching that creepy video. Decay. Decay can start occurring instantly. And it occurs from the action of bacteria and fungus metabolizing the organic carbon from the dead tissue. This is the chemical equation here. And this is the sugar and oxygen. So this is C6H12O6, right, with the common denominator CH2O. So carbon and oxygen will produce water and carbon dioxide. So some factors that will affect the decay is the amount of oxygen. More oxygen means more aerobic bacteria. It promotes faster decay. Higher temperature, warmth, promotes faster decay. So not to be gross, but this is why funeral homes, mortuaries, there we go, mortuaries are so cold. pH, lower pH promotes faster decay. So the more acidity, the higher decay. Humidity, higher humidity promotes faster decay. So in an area like Florida with a lot of higher temperatures and higher humidity is a terrible place to get more fossils. Deserts are great places to start forming some fossils. Better, I shouldn't say great. So this is the sheep bog. This became extremely famous. It was disgusting. So sorry. Where whatever was in this water was actually really good at preserving soft tissue. The sheep's back was exposed. And look at that. It's completely decayed away. So decay is going to be reduced when it's colder. Usually when pH is extremely high or low. I believe this is extremely basic water, but I might be wrong on that. Burial happens quickly. Abrution, or conditions are dry. So burial happens quickly. Abrution, that means a storm event. Those are abrution and storm event are synonyms. Or conditions mean that there's low or no oxygen. All right. So extremophile bacteria does exist. We've learned about that. The archaeobacteria. Actually, nope, that's the next lecture. You will learn about that more. Mostly archaeobacteria exist that may still allow decay, but the reactions are much slower. So some of the ones that we'll look at are sulfur-reducing bacteria here in just a second. Transport. Organisms will often be moved from the place where they die by water. This can result both in great and awful things for the fossil record because water is really where primarily many of our fossils are going to be preserved in the first place. But this can often result in disarticulation, coming apart at the joints, because the soft tissues will decay away. it's almost impossible to get any form of soft tissue in the fossil record. It does happen, but it is extremely rare. Most of the time it's because of really unusual events, or ice or something like that. So here's one way that things are transported. This is called the Bloating Float. And yes, this is an absolutely real scientific term. So what happens here, this is an Ankylosaurus dinosaur, all right? A thyreophorine, that's the larger group name that includes Stegosaurus and Ankylosaurus, and some other groups as well. And when it dies, it liked to spend a lot of its time near water. So when it dies, a lot of the times it would fall into a river or something like that. Well, gases fill a body after death, so it bloats. And when you have something filled with gas that's bloated, often, as you guessed it, it might float. So a lot of our Ankylosaurus friends were actually found in shallow marine sediments or water in sediments. So at first people were like, okay, is this swimming? No, it was actually just bloating and floating after death. This is one of the reasons ammonites are so great at being fossils, index fossils, because after death they would fill with gases in their chambers and they would float around for a little while before depositing. So we would get global ammonites from a very specific snapshot in geologic time. So very helpful for us. Bioerosion. This is a boring sponge. I'm not knocking the sponge here, but it's actually boring tiny holes into a bivalve. This is called Cleona. That's the name of the sponge. This is what it looks like after it was attacked by a boring sponge. There have been holes drilled all into this. Now, they're not doing it to eat the inside of the organism. They're actually doing this so they have a nice home to live in. As you can imagine, though, probably not pleasant for the bivalve. All right, so they will do this into rocks and logs as well, but we very often see this in the fossil record. You very likely have examples of this in your fossil kits. Dissolution. Acidic water will dissolve carbonate shells. That's why Florida is the land of the sinkhole, right? And Disney World. So calcium carbonate, CaCO3, dissolves in the presence of acidic water, which we know from our physical geology class when you were able to dump a whole lot of diluted HCl on top of the limestone and watch it fizz, the great joy of all physical geology labs. So here is a snail shell. It's made out of aragonite, and I know that because all snails are made of aragonite, and it will start to dissolve quickly, just after 2 weeks. So this happens in the ocean continuously because the further down in the ocean we go, the more acidic the water gets, right? So you learn from your SED-PET class about the CCD, the calcium compensation depth, where after a certain point, shells just dissolve because the water is too acidic. Burial. Specimens will be buried by sediment. That is how it works if it is to be a fossil. The weight of the sediment will usually flatten the specimen, or it will be distorted, which is the great sadness to many, many, many scientific experiments that rely on fossils to not be squished. Tectonics, bending, folding, etc., can also distort them. So here's just a summary of what can happen. We can have disarticulation, we can have fragmentation, abrasion, being worn down. So let's say that shell was being bounced along a riverbed here in C. As you can imagine, it'll start to get rounder over time. So then all of a sudden we can start using this just like we can use regular sediments like quartz grains. Say the more rounded, the more it's been worn down. Bioerosion, so things like boring holes. We can have corrosion and dissolution. You should be able to see almost all of these in your fossil kits, including flattening and diagenesis. All right, so here's another example of the potential pathways that we could take here. We have our dead organism. How sad. Becomes a potential body fossil as opposed to a trace fossil. Sometimes we can have complete preservation, extremely rare, or we can have only hard parts left. Once it's burial, we can have it preserved as unaltered, same material. We could have it recrystallized from aragonite to calcite, the more stable form. Material can be removed or added. So all of these different things can leave us with a fossil. So moving into chemical diagenesis and preservation. The first type of diagenesis we'll talk about here is recrystallization. This is a bivalve here. All right, we notice there's a lot going on with bivalve. This bivalve particularly, we see it is, we've got some large calcite crystals in here. You know it's calcite because it's got that gorgeous 6120 cleavage, and you can see some rainbow high refraction in there. Okay, what else is in this image? Well, these are poo pellets. How very fun. So what would you call this in folk and Dunham? I'll let you think about that. Let me know what you think. So we know because it's a clamshell, it was aragonite in life. However, it has clearly been recrystallized to calcitic spar, which you can see. Oh, I gave you the name of the cement. Sorry, everybody. All right. So calcite is more stable than aragonite, and aragonite will often dissolve in the water undersaturated with CaCO3. The calcitic or the CaCO3 rich water will then precipitate calcite into the voids left by the dissolved aragonite, giving us this gorgeous fossil. Replacement. Sometimes we can have a complete replacement of an organism's structure with an entirely different mineral. This is Florida State fossil, agatized coral. All right. So this is coral, and because I know a little bit more, I know that this type of coral, this is a recent coral called scleractinian coral that lived from just after the Permo-Triassic to recent. That's all coral you see today. We know that it is made out of aragonite. This had the aragonite dissolved, and it was completely replaced with silica. You can see this is silica. It's got that lovely sugar grain texture of silica. We also have a special type of replacement, pyritization. So this is an ammonite. Ammonites are made out of calcite. They're made out of aragonite. Oh my gosh, now I'm blanking. They're made out of CaCO3. Okay, we can definitely say that this is different than pyrite. So in anoxic conditions, meaning low oxygen, remember pyrite can't form in high oxygen conditions. We learned that in sedimentary petrology and history of life when you learned about the Great Oxygenation Event, meaning when we know oxygen filled Earth a little bit more because all of our banded iron formations and pyrite deposits pretty much stopped forming. So in low oxygen anoxic conditions, sulfur-reducing bacteria produces hydrogen sulfide, all right, which reacts with iron in the water. And that forms FeS2. That is the chemical formula of pyrite. So if you find something like this, you know it happened in an extremely low oxygen environment, which can be really helpful for us understanding paleo environmental conditions. So again, replacement is when we have one mineral being completely replaced by a completely different mineral. Recrystallization is going to be one polymorph to a more stable polymorph. Permineralization is another specific type of replacement. And this, I want you to know the name permineralization too. The most famous example of this is, as you're looking at it, petrified wood. So how this works is a tree will die. As the tree dies, it lays down in the forest, of course. A lot of the times, mineral-rich fluids, probably rich in silica, will come rushing through, rain and things like that. As it happens, the plant material will be washed away. And as that's happening, the mineral-rich fluid infiltrates the voids in the structure. The minerals will precipitate out to fill those voids, which is why we see the bark on the outside, but the entire thing is now silica. It's usually identified by the gorgeous rainbow color and by the bark pattern. See if you can find the petrified wood in your box. Concretions, you have one of these in your box as well. These are really cool. So this is happening pretty much because we have more iron in the sediment somewhere. So it will happen when the sediment comes really concentrated in calcite or siderite. Usually, even if it's not siderite with the iron carbonate, there is more iron in the surrounding area. But this happens because the organisms themselves are a source of calcium and iron. Those minerals get leached out, leaving behind an impression of a fossil that's usually extremely highly detailed. All of these are incredibly gorgeous plant fossils here. Find the concretion in your fossil box. Now, if you become a geologist, there is about a 99.9,000% chance that at one point in time, someone in your life will come to you and say, "Hello, I found a dinosaur egg," and you'll have to say, "No, is it a concretion?" They look like eggs. It's a really common mistake. It's a really easy mistake to make, but they are certainly not. So what you can do is you can crack it open with a hammer, and you will usually find a beautiful little non-dinosaur egg fossil inside. Phosphatization. So these are a really particular type of fossils. They're not entirely common, but they lead to the preservation of exceptional soft tissues. So this is more likely to happen when pH and oxygen concentrations are extremely low. This is one of the most famous examples of phosphatization. These are from the Doshanteau Formation, which are some of the earliest examples of animal embryos that we have, and they are absolutely beautiful. They're also tiny, teeny tiny. So a preservation bias might see that these are difficult to find, even if they might be numerous. We really don't know at this point. Probably not, because there's a lot of soft tissue. But they're tiny. So they were only recently discovered by Xiao and Knoll in 2000. So very few individuals ever get fossilized. 99.9% of things on Earth do not get fossilized, ever. Which definitely means that there's animals we will never know about because there is no fossil record for them. Which is a really sad thought, but we do have a lot of beautiful fossils, so don't get too down about it. Here's a gorgeous dinosaur fossil here. This looks like an ankylosaur type dinosaur. We can see all the armored plating around its face and on its body. And this is a beautiful fossil with soft tissue preservation even. As you can imagine, this is extremely rare. So when we do find these fossils, they are a true gift from the earth. And when we do find these rare fossils, they're often found in Lagerstaten. So Lagerstaten is plural, Lagerstate is singular. This is a German word, and I'm not going to quiz you on your German grammar here, but what we know is that that Lagerstätte represent incredible fossil preservation sites. They often preserve soft tissue as well as our skeletal material. So here's Hallucigenia and Anomalocaris. These are two incredibly famous fossils from the Cambrian. And one of the most famous Cambrian Lagerstaten is the Burgess Shale. Beautiful. It is a world preservation site, I believe, so you are not ever allowed to collect in there without a lot of scientific permits. But Hallucigenia, sorry, I'm going to go back here. This has a pretty fun story. The person who found this really honestly thought that he must have been hallucinating, or it looks like something you would see if you were hallucinating, which is where it gets its name from. Like, what the heck is this animal? So here is one of the most important scientific questions of all time. If Hallucigenia wore pants, would she wear them like this, here on the left with the pants on the bottom, or on the right, pants on top? We may never know the answer to this question. So again, Lagerstätte is plural. Here is the Jholebiota. This is a famous Lagerstätte recently discovered of China that shows feathered dinosaurs. Look at this baby. This is Sinusoidal X. Look at the little baby feathers. This is extremely rare. Fossils like this have raised new ethical dilemmas in fossil study because people have found out that if they find these fossils, paleontologists will pay top money for them. I am not knocking anybody who does this, please believe me. It just raises ethical dilemmas because the rise of fake fossils on the market has actually been rising. So there are questions that scientists have to answer about ethics when buying fossils. And we'll talk more about that in a couple weeks. Here's a close-up picture of those fossils. These are so good we can actually get chemical traces out of them. And we've actually been able to determine that they match very closely with the chemical composition of bird feathers today. And there is a cute little drawing of Sinusoidal X. So what biases do you think might be introduced into the fossil record? So many, right? So let's talk about these biases. The fossil record is so biased and so incomplete, but it's not so biased and so incomplete that we can't use it. We just need to understand what those biases are. Most organisms, the large majority of organisms, do not get fossilized. There are massive gaps in temporal coverage, thanks to depositional and unconformities, subduction, fewer older rocks. Remember, the oceanic crust recycles itself about every 200 million years, so we lose all those fossils too. Environmental coverage, thanks to ecological and preservation bias. So marine environment, shallow marine, much more likely to be preserved than rainforest. All right, remember rainforests are high humidity, high temperature. They're not as easily to get buried because ocean sediments usually bury fossils much faster. And anatomical preservation. So fragile, soft-bodied organisms are super hard to preserve. Pterosaur fossils are incredibly rare. And unfortunately, one of the greatest tragedies in modern paleontological times, the Brazil fire that burned the National Museum, almost 20% of the world's fossils were lost to that fire of pterosaurs. So heartbreaking, absolutely heartbreaking. Always support your natural museums. So preservational biases here in environmental. A lot of environments are better preserving fossils than others. For example, shallow marine shelves are great. They bury things quickly. If you're buried, there's going to be less exposure to oxygen, things like that. Rainforest is terrible. Most of the time, it'll just sit out and decay until it's gone. So we don't have nearly as good a handle on what rainforests look like as we do our shallow marines. Some animals are just more common. We have tons more fish fossils than sea turtles. Sea turtles are not nearly as common in the fossil record. They reproduce at much lower numbers. Some animals are just much more fragile than others, and they're more likely to be smashed in transport. All right, so here we have our example here. Bird bones are hollow, meaning they're extremely delicate and they'll get smashed. So the coyote here is much more likely to fossilize, even if the worst is about to happen to him. Collector bias. These are occurrences of foraminifera throughout the Phanerozoic. What do you notice? If you're noticing that there is an overrepresentation in North America and Europe, you would absolutely be correct. We have much more of that. There's another collector bias in that fossils are much more likely to be found if you're within a couple miles of a museum or a university. So there's a lot of different collector biases here. Also, another collector bias is size. If you're out in the field, are you going to pick up a teeny tiny little thing you might see? No, you're going to pick up the stuff that you can see. There's all kinds of collector biases. And as a reminder, we've talked about this already, but why would there be an overrepresentation of fossils being found in North America and Europe? That would be a collector bias relating to the inequities present in all fields of science today, where scientists work much more heavily in North America and Europe and do not collaborate nearly as much with scientists in other parts of the globe. So that's something we need to change. So is the rock record driven? Is the fossil record driven by the rock record? Biodiversity in the fossil record is correlated with the volume of sedimentary rock, meaning a lot of the times we have more fossils when there's more rock. Make sense? Now the volume of sedimentary rock is correlated with sea level, meaning higher sea levels, more rock. Lower sea levels, less rock, and therefore less fossils. So could there be a bias in that whenever we see fewer fossils, we usually assume a mass extinction? Could it just be that there's less rock? We don't know. There are a lot of questions. We'll talk more about this bias throughout the rest of this course, but it's possible that some of our mass extinctions are actually influenced by just what's preserved. So think about that moving forward. Now, plate tectonics do affect biodiversity. So when we start to break up these ocean basins here, like in the late Permian with Pangea, we start to get teeny tiny little ocean basins between all the continents, which actually drive biodiversity because we have a lot of speciation occurring. But there's a lot to talk about with this story, so this is just an introduction to this particular point. Another bias, Lazarus taxa. Those are those species that go extinct and reappear later. We think that they go extinct, So the coelacanths, of course, we already talked about this one. We thought they died in the KPG, but it turns out they were living happily in the Indian Ocean, and we found that in 1938. Trace fossils. This is a great one to talk about here. Body fossils and trace fossils. Body fossils are the body themselves. Traces are whatever they've left behind. So we find footprints, burrows, predation marks. Here are some cut marks on bones. We have footprints here with ground ripple marks. We have poo, coprolite. All of these are evidence that the animal was there, and we can learn a lot about behavior from these trace fossils. However, unless the animal happened to die in the middle of making that track and it's left behind, it's extremely difficult to infer which species made the track. This is because, obviously, species make a whole lot of different tracks depending on what's going on. If you, the human, dug into the sand with your hands and then made footprints walking away from the beach, it might be difficult to figure out the same person made those motions. So thought question, the pull of the recent. This is an idea. Actually, I want to go back here one more time. Sorry. Also think of another bias here. How many footprints can you make in your life? A whole bunch, right? So if animals are more likely to leave behind tracks, could that possibly influence our thought of diversity in the fossil record? As far as I know, you only have one body, but that one body can make a bazillion different footprints. So think about that when we're thinking about bias in the fossil record. So the pull of the recent is the idea that we have more sedimentary rock from the Cenozoic and more recent, even though we actually have a heck of a lot less by percentage. So the Cenozoic has only been going on for 65 million years, and we don't have that much rock volume, but we have way more fossils from the Cenozoic than any other time. So that's the pull of the recent. How might that bias the fossil record? You tell me. Here we have the pull of the recent and in a very different way. So this is diversity here. The higher this line, the higher the diversity. How might the pull of the recent be a bias? So this is a famous biodiversity curve. It's based on the number of marine families. Families, what are families? This is Linnaean taxonomy. So think about this curve. Think about how these biases might be introduced here. We're measuring biodiversity through time. So if I were to say, I don't know, give you this graph on an exam, perhaps someday, maybe in a couple weeks, I don't know, this is hypothetical, I might ask you to explain biases that might be introduced using this curve. So how might the Cambrian explosion be influenced by taphonomy? How could it be influenced by mineralization? The Cambrian explosion is when we start to see the first types of hard shells. So is it an explosion of life, or was it an explosion that we could just see for the first time? The Cambrian explosion was very likely an explosion of life, yes, but some of that is also a taphonomic artifact in that we were actually able to see this for the first time. What types of diversity were you not easily able to track? Well, some of the most diverse things. Beetles have a terrible fossil record. Worms have a terrible fossil record, and those are both extremely diverse groups. So think about that when we think about what we see in the fossil record. What are we missing? What windows can we not see through? Here are your review questions. I will see you soon for the next lecture.